Hello, my name is Koen Beerens from the UGent Center of Synthetic Biology, and in this presentation I will explain to you how we use enzyme engineering to enable the production of rare sugars. We all have already eaten carbohydrates today, either in the form of sugars in a cookie, candy or piece of fruit, or in the form of a fiber, like starch in bread or pasta. Furthermore, we all have heard of the negative connotation linked to carbohydrates and excess consumption thereof, leading to obesity and diabetes. However, carbohydrates are much more than just a source of energy. Indeed, they mediate a wide range of recognition processes that are central to human health. Hence, carbohydrates find a wide range of applications in, for example, the food and pharma industries. Examples are prebiotics that stimulate the immune system and low-caloric sweeteners that can help to control the effects of diabetes. In addition, carbohydrates are also important building blocks for active pharmaceutical ingredients like antiviral and anti-cancer molecules. Unfortunately, the most interesting sugars or saccharides are scarce in nature, making it too costly and inefficient to extract them from natural resources like, for example, honey. Therefore, we aim to develop biocatalytic processes to make available those special carbohydrates, also known as rare sugars, from cheap and abundant substrates like, for example, table sugar, chemically known as sucrose, or starch and cellulose. Unfortunately, carbohydrate synthesis is tricky due to the multiple reactive hydroxyl groups present, as you can see here in the example of glucose, the most abundant monosaccharide. As a clear example that the world of carbohydrates is very complex and its synthesis routes very challenging, we will show you the example of coupling two glucose molecules to each other. This can lead to no less than 11 chemically diverse disaccharides, each with their own characteristics. Hence, it is clear that there is a strong need for the control of regio and anomeric selectivity. To do so, we fall back on enzymes as natural biocatalysts because they allow the synthesis of rare disaccharides with high selectivity. At first, I would like to give you some background on the target sugar that I will be discussing here, namely Kojibiose. This is a rare disaccharide found in sake, beer and honey, but only in very, very low quantities, making it very expensive as a rare sugar. Nonetheless, earlier research had already indicated that it could be an interesting sugar to produce since it displays promising properties like not cariogenic, meaning that it does not promote tooth decay, it is lower in calories, and it is potentially prebiotic. However, this evidence is still superficial, mainly due to the fact that further research is limited due to the low availability of Kojibiose. Therefore, we aimed at producing this interesting rare sugar at large scale using biocatalysis. Our main working horse as a biocatalyst is an enzyme called sucrose phosphorylase, abbreviated as SP, which in nature performs the phosphorylasis of sucrose to form glucose 1-phosphate. However, more importantly and interestingly, in the absence of phosphate, this enzyme can be used as a transglucosidase to produce glucosides, starting from sucrose or table sugar as a glucose donor. In case the acceptor molecule is a monosaccharide, the reaction product will be a disaccharide, which is what we aim for in this research. Unfortunately, when given glucose as an acceptor molecule, our sucrose phosphorylase enzyme makes two products. Approximately 2/3 of the product is maltose, a sugar that is also obtained in the malting process of beer making, and approximately 1/3 Kojibiose, the rare sugar of interest. The lower selectivity is unfortunate, but not abnormal, since we are exploiting a side reaction of the SP enzyme. Therefore, we will need to apply enzyme engineering to increase the selectivity towards the formation of Kojibiose. Enzyme engineering is a technique in which we will alter the DNA encoding for an enzyme or a biocatalyst in order to change its properties like selectivity or stability. After mutagenesis, the DNA will be transformed typically into bacterial cells for expression at small scale, allowing us to process multiple variants in parallel and screen for variants with desired properties. In a typical enzyme engineering study, we tend to saturate so-called hotspots in the enzyme's active site. In this case, these hotspots were residues that are in contact with the substrate as shown in the central figure. Saturation mutagenesis means that we will change the residue of the hotspot by all 20 amino acids, creating a library of roughly 100 variants that needs to be screened. In this specific case, around 10 hotspots were targeted, and repetition of the process on beneficial mutation and combination of beneficial mutations was then performed in search for further improvements. For this, we now also have available a molecular biology robot that can help with the automation of most steps, drastically increasing our enzyme engineering throughput. The created libraries were then evaluated using our HPLC system specialized for carbohydrate analysis by focusing on both improved selectivity and activity. Here you can see the results of the semi-rational enzyme engineering of SP for COGI-BIOS production. The circle in the middle, marked with the two lines, is the wild-type enzyme, producing approximately 33% COGI-BIOS with a certain amount of activity, which is less important at the moment. The most important here is that most of our single-point variants increased the activity in COGI-BIOS formation, and when these were further combined, the selectivity further improved to no less than 95%. In the next step, multiple variants were then evaluated to find the best production process conditions, and the best variant and conditions were used in a large-scale production. In simple laboratory equipment, we performed a production at 10-liter volume, using the best SP variant under mild conditions, being pH 7 and 55 degrees Celsius, yielding over 5 kilograms of COGI-BIOS in solution. Via a simple yeast treatment and cooling down crystallization, we were able to obtain 4 kilograms of highly pure COGI-BIOS. Our engineered SP variant and the optimized biocatalytic process have the potential of producing COGI-BIOS at the cost of only a few euros per kilogram, hereby marking a very important milestone for further research of COGI-BIOS's properties and its future applications. The enzyme engineering exercise was later repeated to produce nigerose, another expensive and rare sugar found in sake and honey. Starting from an already known mutant, we were able to improve activity by 36-fold and the nigerose to maltose ratio by almost 8-fold. We again performed a large-scale production, but were less fortunate since the nigerose did not spontaneously crystallize, showing that the nature of the glycosidic bond impacts the physicochemical properties of the glucobios. Nonetheless, we were able to purify 100 grams of nigerose using our sugar purification system. We later also produced disaccharides that contain monosaccharides other than glucose, such as, for example, galactose, ribose, and arabinose, hereby creating a library of rare disaccharides that can now be evaluated for their respective properties, which can in turn help us in the understanding of the structure-function relationships of rare sugars and their potential future use in, for example, the food industry. In summary, enzyme engineering is a very powerful tool to enhance the properties of biocatalysts, such as, for example, selectivity, as was shown here in this example for the biosynthesis of multiple rare disaccharides. It allowed the creation of robust biocatalysts that could be applied in efficient production processes, resulting in high yields and purities. The available equipment clustered in the high-throughput screening for synthetic biology core facility not only allowed us to successfully execute these challenging endeavors, but it also allowed us to reduce the development time and cost compared to when this equipment would have been absent.